All right, fire away when you're ready, Dilly.
Hello, everybody.
You lead me in.
Yes, hello, everybody. To start off, I'd like to do an acknowledgement of country. And the society, that's the Australian Wildlife Society, acknowledges the traditional custodians of country throughout Australia and their continued connection to land, water, and culture. The Society wants to pay our respects to elders past, present, and emerging, on whose lands we live, work, and all of the studies that we're going to be discussing today were conducted. And I'd particularly like to welcome any Indigenous people with us here today. So I'm Julie, and I'm the president of the Australian Wildlife Society. And today we're celebrating Australian Wildlife Week. And it was established in 2019, and we are celebrating it right across Australia during the first week of October each year. And it's basically to encourage a positive relationship between humanity and nature. The Society aims to raise awareness of wildlife conservation issues and inspire all Australians and people across the globe to explore and develop a deeper understanding of wildlife conservation issues, both flora and fauna, and to gain the necessary skills to make informed decisions and implement wildlife conservation action. So before we move into the actual webinars today, I just wanted to highlight that our wildlife of the year for 2023 is Proteaceae. And we want to really encourage everybody to go and have a look and find out more about this group of wonderful and unique plants. On our webpage, that's aws.org.au, we have a fantastic article written specifically on that group of plants, the Proteaceae, and that's written by one of our fantastic volunteers, Annie Nguyen. So please go and have a look at that link on our webpage. So today, we're going to be talking about a wide range of topics, and we have lots of keynote speakers and 6 early career researchers, which the Society is very, very proud to support. And this year we are showcasing one of the recipients of our very first Dr. Clive Williams Memorial Scholarship. But as well as that, our other speakers have also been successful in obtaining funding and grants from the Society. So just before we begin though, I just want to make sure that everybody's video and audio, it'll be muted, okay, just to make sure that the speakers don't get any interference. And a member of the Society is going to introduce each speaker. And if you want to submit any questions, we have a little Q&A section, and the keynote speakers will have some time at the end of their talks to answer those questions. As well as that, we have the chat, so feel free to maybe put in the chat something about where you're viewing this from, and your interest in Australian wildlife, either flora or fauna. So we've got the chat open there as well. So at the end of the keynote speakers, our manager, Megan, will be asking the floor if there are any questions for those keynote speakers. But the shorter talks will just be responding in the Q&A to questions and answers. This webinar is currently being live streamed on Facebook and YouTube. And after the event, we're going to pop up the recording on our website and on YouTube as well. So if you want to revisit it, or you've got some friends and family that would like to have a look at the recordings, please go to those sites and have a look. So without further ado, it is my very fantastic pleasure to introduce our first speaker, Jack Bilby. He's from the University of New South Wales, and he is our very first Dr. Clive Williams OAM scholarship recipient. Now, unfortunately, Jack is in the field, so we have a pre-recorded talk from him today, so he won't be able to answer any questions. But if you do, please pop them in the Q&A anyway, and we can pass them on to Jack and he can answer them at a later date. So Megan's going to get his seminar up, but he's going to be talking about bandicoots and how they respond to extreme heat in burnt and unburnt habitats.
Can you hear that? Here we go.
Here we go.
Hello everybody, my name is Jack Bilby. I'm a PhD candidate at the University of New South Wales, and today I'm going to be talking about a piece of my PhD research, which is how do bandicoots respond to heat waves in burnt habitat. Firstly, I'd just like to acknowledge the Bangala people on whose land my fieldwork is conducted, and also the Ghana people on whose land I'm on today. So extreme heat events are increasing in duration, frequency, and intensity, which is quite alarming for ecosystems across Australia. So the arid zone in particular, which is already subjugated to high temperatures, is of particular risk, as these areas are projected to have the greatest increase in heatwave intensity. And yet we know very little about how arid zone species actually respond when temperatures get really high. So my study species for this project is the Shark Bay bandicoot. So this is Australia's smallest bandicoot species, and it's one that has a similar story to many. So they've been pushed to the brink of extinction by cats and foxes and have been reintroduced to many areas in their former range. But we also suspect that this species is at particular threat due to extreme heat events, due to their small size. They only get up to around 250 grams, which means they heat up very quickly. And also the fact that they nest on the surface. So you can see there in that second photo, we have a bandicoot nest comprised of sticks, leaves, and other little bits of debris. And generally these are positioned under shrubs or under the canopy cover, but what happens in burnt areas when those things get completely wiped out? So that's the main goal of this research. So to track the bandicoots, we put tail transmitters on them. So these enable us to put VHF transmitters, which mean that we can actually track the animal to its daytime shelter site, see exactly where it's sitting without having to disturb it. And then using GPS transmitters as well, we're able to see how far the animal's moving at night, what habitats it's using, and do that all remotely. So you can imagine that a bandicoot in an unburnt area will have plenty of canopy cover, plenty of material to build a sturdy nest, and this will help just mitigate some of the worst of the ambient heat. But once the fire's gone through, you have substantial reduction in that canopy cover, and so you have much higher surface temperatures due to that increased solar radiation exposure, and we suspect this will drive increases in nest temperatures as well. What this might lead to is decreases in the activity window that bandicoots can actually forage during. So you might find they're only able to forage during the coolest part of the night after they've actually cooled down from the hot day, and you might therefore find decreases in fitness and survival. So my study site is Secret Rocks Mallee Refuge on the Eyre Peninsula. The reason for this is the fire history of this property and the fact that bandicoots have been reintroduced there. So a substantial amount of the habitat that was occupied by bandicoots was burnt in the 2019 Black Summer bushfires. As you can see by that dark grey outline there, it covered a substantial area of the property. And there was also a small controlled burn back in 2013. But despite substantial changes to the vegetation structure of these burnt areas, there are still bandicoots utilising them. So that kind of brings about the question, how are they surviving and what are they doing when the temperature gets excessively high? So I'll share a bit of preliminary data from last summer. We'll start with looking at this landscape here, the area highlighted by orange with a thermal drone. So looking at this, you can straight away see the lighter red areas and less purple areas where the burns have occurred. So if I just outline them quickly, you can see that inside the area with the thick black line, the 2019 wildfires, there's very little canopy cover shown by the darker and cooler purpley blue areas. But you can also see that in the 2013 bushfires, 2013 prescribed burn, I should say, there's still reduced vegetation through there, causing higher surface temperatures. And we can see when we look at bandicoot nests within the unburnt and burnt areas that this same trend follows. So you see much higher canopy indicated by the solid colours in the unburnt compared to the burnt areas. Now, we've also trialled putting temperature loggers, eye buttons, into different nest types, but we were quite limited last summer by it both being quite a cool summer and limited by the amount of data loggers that we were able to actually obtain. So we haven't been able to detect any notable differences there. You can see quite a few gaps in that research still. Thankfully, the grant money that we've received from the Australian Wildlife Society will go towards ensuring that we have enough data loggers for the hot summer that we're forecasted to have ahead. Just like to say thank you all for listening, and thank you again to the Australian Wildlife Society for supporting this project through both the Student Research Grant and the Clive Williams Memorial Scholarship, and a big thank you for letting me share this research with everyone tonight. If you have any questions, don't hesitate to reach out to me via social media or email. Thank you.
Okay, thank you. Over to Brian to introduce our next speaker, please.
Hi, my name is Brian, and I'm the director of the Society, a director of the Society. Our next talk is also a pre-recorded talk from Koala Smart, who will share with us their work on engaging children in conservation-based lesson plans.
Hi, my name is Lynn Wallace-Greenberg. I'm an environmental scientist and a conservation ecologist working with Koala Smart, an educational resource designed to engage students and their teachers to learn and impart their knowledge about koalas and their habitat. Koalas in New South Wales and Queensland have been listed as endangered following the catastrophic bushfires of 2019-2020 that killed or displaced approximately 3 billion mammals, birds, reptiles and frogs. Of these 3 billion, approximately 60,000 were koalas. Bushfires, along with environmental and climate change pressures, may bring about the extinction of koalas in New South Wales and Queensland by 2050. However, with knowledge and the willingness of scientists, educators and the concerned public, we can avoid this drastic fate. We see the future of endangered species such as koalas to be in the hands of our young citizens. Koala Smart has been developed to build awareness and develop skills in students so they are confident in taking positive action to address declining koala populations. As educators, you are instrumental in saving koalas from extinction by teaching, motivating, and developing your students' knowledge and skills. Together, we can change the trajectory of our koalas and ensure that they survive for future generations to enjoy. Koala Smart emerged following Koala Conference in 2018 as a collaboration between the Tacking Point Lions Club, the Port Macquarie Koala Hospital, Hello Koalas, and a koala ecologist from the mid-north coast of New South Wales. It was established that there was a need to impart knowledge to the wider community about the threats that both koalas and their habitats are facing. So it was decided that the best way to disseminate this information was to connect with students through their teachers at local schools. Since its inception, Koala Smart has been building resources for students and their teachers. We are proudly supported by the Koala Program, a division of the New South Wales Department of Planning and Environment, and by Koala Conservation Australia through the Port Macquarie Koala Hospital. The Koala Smart platform is a free, curriculum-based educational tool that assists both teachers and students. It is designed to actively engage students in the conservation of koalas and their habitat. Students can then take this knowledge and become involved in hands-on projects that inspire them to find solutions to conservation challenges, thereby helping to ensure the koala's survival. Teachers can easily access these free resources by going to our website, www.koalasmart.org.au, and then under the Resources tab, you can register. Upon approval, the teacher can access 6 lesson plans, including worksheets, activities, educational videos, and interviews with experts. All of these resources are electronic and can be downloaded or printed for ease of use. Teachers also have the opportunity to engage in projects with their students and can receive guidance in the form of incursions or excursions with a critical friend. Our critical friends include environmental and conservation professionals, volunteers, wildlife sanctuaries and hospitals, arborists, rangers, and koala rescuers. After a teacher has registered, their students can access the students section of the website, which will enhance their conservation journey. The lesson plans provided can be used in their entirety or in any combination that suits a teacher and their students. Importantly, all lesson plans are aligned with the New South Wales, Queensland, and Victorian school curriculum. These lesson plans are currently being modified to allow teachers to easily access age-appropriate information for their students. Lesson 1, Community and Social, investigates the koala's home range by looking at the community structure of a koala population. It investigates how habitat loss impacts the koala population, affecting the genetic diversity and ultimately the survival of the koala. Lesson 2, Eco-Biosystems, healthy trees for healthy koalas. In this lesson, we look at the key threats to koalas and discuss how important trees are to the survival of healthy koalas. Lesson 3, Forest Spirits, the Heart of the Bush. We explore the spiritual link that the koala has with our First Nations people and look at creative and artistic ways that students can raise awareness of the challenges that face the koalas through artwork. Lesson 4, Our Planet, Ourselves, our connection with koalas. Here we explore adaptations that assist koalas to survive in their environment. We also look at ecology, how the environment is directly linked to species survival and the koala's overall health. Lesson 5, Desirable Futures. Protection for now, protection for tomorrow. In this lesson, students are given the opportunity to plot a realistic course of action in a fictional setting that will protect koala habitat. Lesson 6, Champions for Change. Shout it to the treetops. In this lesson, students can create a storyboard, a podcast, or an article that discusses koala conservation in an informative and persuasive manner that can inject change. Upon completion of the lesson plans, there's the opportunity to create a unique school project. While Koala Smart offers guidance for projects, this is an optional extension of the program, and choosing not to engage in a project does not impact the ability of teachers and students to fully enjoy the program. A Koala Smart project is successfully showcased by students from St Columba's Anglican School in Port Macquarie. These students were distressed that land clearing next to their school was destroying known koala habitat, and so they set about rectifying this situation. They contacted the Port Macquarie Koala Hospital for advice on which trees to plant. The Port Macquarie Koala Hospital supplied seedlings of swamp mahogany and red gum, which the students planted around their school. They've also helped stop urban development in Ruins Way, Port Macquarie, which is now designated as protected koala habitat. But they haven't stopped there. They have future plans to build koala access ramps so that the koalas can easily reach their feed trees in the school grounds. The goal of Koala Smart is to ensure the survival of koalas in the wild, so their predicted extinction by 2050 does not become a reality. To achieve this, we need a community approach and rely upon our scientists, ecologists, politicians, and our teachers to lead and motivate tomorrow's leaders and citizens to actively engage in the protection of the koala and their habitat. With your help, we can arrest the decline in koala populations together and achieve this goal. There is a short video from the students of St. Columba's Anglican School where they speak about their actions to save local koalas. You can access this video by going to our website www.koalasmart.org.au and then under the About tab, select News. This short video is called Students Step Up for Koalas. Will your students take the challenge and step up for our koalas? Their future is in all of our hands.
Okay, a very great presentation there from Koala Smart. Thank you, Julie. I'll get you to introduce the next speaker, please.
Yeah, that was a really good talk about koalas. I find it really interesting that just last week, actually, I saw a koala on the road, and it was quite distressing when not everybody was slowing down. So it's really important to get that message out about, koalas and other wildlife on the roads and to drive carefully. Okay, so our next speaker is Paula Ruiz from the University of Tasmania, and she's going to be talking about the mechanisms of stability for degraded turf-dominated reef states. Thank you, Paula.
Oh, thank you very much for let me present here. So I'm going to share my screen right now. Okay, and good. So hello, everyone. I would like to talk to you about the mechanisms of stability for degraded turf-dominated reef states. That is basically my PhD project. So I would like to talk to you about the reef states that basically in Australia, we have two different reef states in tropical and temperate reefs. For tropical reefs, we have as a stable state the coral reefs, while in temperate reefs, we also have the kelp forest, that both of them are the food and shelter of the marine ecosystems. But we also have some unstable states like the collapse of coral reefs or the urchin barrens, especially here in temperate locations like Tasmania. So what happened after a perturbation? We also have the collapsed ecosystem, but in those ecosystems, we have the turf. But what is the turf? The turf is basically different algal mats that are composed with filamentous algae that can trap sediments, and these trapped sediments are the mechanisms that prevent the recruitment of habitat-forming species such as kelp forest or coral. But also turf can dominate the intertidal and subtidal rocky coast in global scales and tropical and temperate reefs. Those algae are first colonizers and also can rapidly grow in nutrients enrichment and sediment-landed environments. And it's also very easy for them to colonize available space in degraded environments. That's why we have these turfs in all the eastern coast of Australia. So for my PhD project, we have two different questions. The first one is, which is the best location for turf to grow? So to answer this question, we are deploying some underwater experiments with sediment panels, and Lizard Island, that is the beginning of the Great Barrier Reef, as well in Heron Island, that is the last part of the Great Barrier Reef. But also different locations around Tasmania, such as Bigelow, St. Helens, Fortescue Bay, and others, that will help us to understand what is the mechanisms of turf and what is the persistence of these filamentous algae in tropical and temperate reefs. So to answer these questions, we deployed some different sediment panels in tropical locations and in temperate locations, and we left there for 2 or 3 months in order to understand which is the best location for turf to grow. But also, we want to understand if this turf can persist in these locations. So that's why we also deployed some underwater experiments. Like we can see in these pictures, we just split the sediment panels with turf coverage, and we put it in collapsed and healthy patches around the different locations in tropical and temperate reefs. So for the tropical reefs, we deployed the sediment panels in healthy locations such as the coral reefs, but also the collapsed reefs, as we can see in these images. And for temperate locations here in Tasmania, we deployed some sediment panels in kelp forest, but also in urchin barrens to understand which is the best place and how long can persist the turf in those areas. So what's next? Because of the grant that the Australian Wildlife granted to us, we can understand also other locations such as Sydney, that it's like a subtropical place. So because of this, we can deploy some underwater experiments in places such as Little Bay, Bear Island, and Shipwreck, and others around Sydney, in order to understand the mechanisms not only in tropical and temperate locations, but also in subtropical locations. So thank you for letting me be here and present my PhD project. And if you have any questions, please let me know.
Great, thank you so much, Paula. That was fabulous. So if anyone has any questions for Paula, if you can please just pop them in the chat, and no doubt she'll be more than happy to answer them for you. So I'll just ask Dr. Hayley Stannard to introduce our next speaker. Thank you.
Megan. Good morning, everyone. So I'm Hayley, and I'm one of the directors on the Australian Wildlife Society board. And I'm delighted to introduce to you our next speaker, Nancy Pratt from Pioneer Catchment and Land Care Group. Thank you very much, Nancy.
Thank you, Hayley. I'll just share my screen. All righty. So good morning, everyone. My name's Nancy. I'm the projects coordinator at Pioneer Catchment Land Care. Our organisation is situated in Mackay, which is along the Queensland coast, just south of the Whitsunday Islands. We undertake land care activities from the Great Barrier Reef and its many islands to our east, and over 100 k's west to the majestic Yungala region and the northern stretches of the Clark-Connors Ranges. We currently have 5 full-time employees and 2 part-time employees. Our organisation has grown over the last 3 years to include commercial conservation services, school-based education and training, and large-scale land care projects. We're aiming to rely less on grant funding and more on those long-term contracts to keep our organisation stable. Our regional challenge in our area, it's not well documented in terms of regular and usable evidence in terms of our flora and fauna species diversity and the numbers. Locals know about our amazing endemic species such as the koalas, the quolls, platypus, youngler honeyeater. However, our geographical location limits our ability to have year-on-year research projects that collect vital data of our flora and fauna. We unfortunately don't have the university students looking at doing PhD research up this way. We don't have many local experts or ecologists. We don't have the large environmental groups or the number of environmental groups to collaborate with. And our Queensland Parks and Wildlife services up this way are extremely limited in their funding and the number of staff they actually have on ground to undertake any kind of research projects. Because of this lack of data, we are finding it harder and harder to have those statistics, the data to back up our conservation projects, and therefore it's hard to compete against the increasing number of organisations Australia-wide and locally for funding. We know other locations around Australia have years and years of research and data. However, we are only just really starting. So to solve that problem, we've looked at two areas. And the first one is that collaboration between groups. As all you listeners know, collaboration is key, and bringing interested groups together to share knowledge and resources. The only problem with this strategy in our region is that these groups are few and far between, and the ones that we do work with in our region are stretched for funding, experienced and qualified staff, and available time and resources. So our next port of call is turning to our environmental enthusiastic community members and introducing them to the power of citizen science. We need those bushwalkers, the farmers, the peri-urban landholders to take up an active interest in what's in their backyard and have the skills to correctly document their observations and sightings. So this is where our story begins. So only just yesterday, we hosted our first annual Youngler National Park platypus count in the Broken River region of the park. We chose this location and the platypus as our first citizen science event for a number of reasons. Youngler National Park is one of the few places in Australia where the public can be confident of seeing a platypus and is a local tourism draw card. Sites were easily accessible for participants with varying levels of fitness. We surveyed the same sites as the Queensland Parks and Wildlife Service did in 2012, so we had a little bit of comparable data. The original study in 2012 was designed after concerns were raised by locals and tourism operators of material from adjacent dairy farms washing into the park's rivers during heavy rain. The survey measured microinvertebrates and platypus numbers at 3 different sites. Platypus are also a hot topic in our region at the moment, with the state government proposing to construct the world's largest hydro dam just north of the Broken River and bordering the national park. So due to this, we hoped our attendance numbers would reflect the community's concerns. Just to note, the survey areas are a fair distance from the dam's footprint, and the dam is not expected to impact these areas. Recognising the platypus and positively identifying one does not require an expert. So participants were able to survey the presence of platypus in small groups or solo, and our organisation didn't have to go to the expense of flying in and accommodating experts to achieve the objectives of the event. So the event went off without a hitch. It was great yesterday. We had a total of 16 participants join the survey. We introduced them to the concept of citizen science, gave them a general rundown on the ecology of platypus, a step-by-step guide to iNaturalist and the project that we had set up in iNaturalist so they could record sightings outside of the event, and why it is so important to collect this data and what it could mean to the conservation actions within our region. Each participant was given a clipboard with a survey sheet, the locations, and a survey timeframe, and sent on their way. Just some of the, just for the fun of it, I just added a couple of little sightings from a couple of the participants yesterday. So the weather was chilly and overcast, so it may have hindered some of our results. So lessons learned from our first citizen science event. So unfortunately, few platypuses were actually spotted, which could have been due to a number of reasons. The weather, the season, the time of day. Therefore, we're having that discussion to hold the event several times during a year. A few of the locations surveyed nil platypus, which coincided with the Queensland Parks 2012 survey results, and therefore these locations could possibly be dropped off the survey list and other locations added, which may be more suitable locations for platypus. We could encourage private landholders around the region to participate in the event, whether that's allowing participants onto their properties to survey their watercourses and dams, or arming the landholders with their own techniques to undertake survey activities themselves. Volunteers were happy to use just a simple survey sheet and hand it back to us for inputting into iNaturalist. The event was a great start to introduce the concept of citizen science. We understand that getting community members inputting data into nationally recognised databases is going to be a long-term project in our region, but we will continue to educate and encourage our community. We're also looking at survey events of other species in our region, especially those listed as threatened, endangered, or vulnerable. And any help from other groups is always greatly appreciated. And as this is not a new concept, so we're not inventing the wheel. Other organisations already know the do's and don'ts, and we're just starting to catch up really in our region. So yeah, so that was our first citizen science project. Thank you all for listening.
Fabulous. Thank you so much, Nancy. A fabulous talk. And firstly, congratulations on the citizen science event that was held yesterday. We also have Samantha in the chat who says it's a huge challenge to continue with limited capacity of funding. So thanks for thinking outside the square, and hopefully there is some better funding on the way. And I think it's probably a key time to highlight the Society's conservation group grants and awards. So we always encourage community groups to apply for our conservation group grants, which are available on our website, and our annual awards. We've got 5 prestigious awards, and applications for those awards are due on the 31st of December. So we encourage you to apply for one of those.
Thank you.
So that was it for any questions at the moment, but if you can just keep an eye out on the chat or the Q&A to answer any further questions, that would be much appreciated. So for now, we have Patrick, if you could please introduce our next speaker.
Megan, can you hear me?
Yes.
Oh, okay. I thought
Sure.
I was muted off. Thank you. Great to be back. And what a wonderful group of speakers it is. I'm not quite sure the name of the person I'm introducing, but my pleasure to introduce the next speaker, and I hope that we can learn more about Australia's wildlife.
So we've got Anne Ibbotson from the University of Newcastle talking to us about...
Lovely.
Yep.
Lovely. That's our newest, latest scholarships at the university, and it's powering on because we already had some candidates for me already, I believe. Thank you. It's my pleasure to introduce the next speaker.
Thank you.
Thank you, everyone. I'll just share my screen. So hello, everyone. My name is Anne Ibbotson. My background is in veterinary science, but this year I recently commenced a PhD in the Conservation Science Research Group at the University of Newcastle. The Australian Wildlife Society is kindly funding my research project to see whether stress and reproductive hormones can be integrated with ecological measures to improve conservation decisions for the Little John's tree frog, which is this beautiful tree frog you can see here. So today I'll be speaking about this project and my PhD more broadly, which includes measures of immune function and extends to the green and golden bell frog. Frogs are fighting their own pandemic, the fungal skin infection chytridiomycosis, or chytrid for short. It's devastating populations, and we've added human-driven threats such as habitat loss and fragmentation and climate change. The situation is critical. Over 40% of amphibian species are threatened, and this includes the Little John's tree frog and the green and golden bell frog. Unfortunately, further declines are occurring in many species despite conservation efforts, highlighting that new approaches are needed, both to understand the impacts of these threats on health and to also monitor and inform conservation efforts. My project involves developing minimally invasive biomarkers of amphibian stress, reproduction, and immune function to inform environmental management decisions. We know chronic stress has negative impacts on health, and it's important to understand the interplay between stress, reproduction, immune function, and disease, as it has major implications for population health and ultimately species survival. Conservation strategies are in place for my species, including breeding programs and initiatives to create artificial ponds and connect habitats, but biomarkers provide a platform to build on this. I plan to measure the main stress hormone, corticosterone, the reproductive hormones, estrogen, testosterone, and progesterone, and a novel marker of immune activation, neopterin, in urine using mass spectrometry, which is considered the gold standard of hormone analysis in people. I'll then correlate these results with reproductive output and status by measuring egg size based on ultrasound, which are these little white things that you can see on the screen, and sperm quality in males. I'll then also correlate those results with genetic diversity as a level of, as a measure of inbreeding, chytrid status and ecological measures, which should allow us to compare populations and also look at the relationship between chytrid and reproduction and stress levels in these frogs. Another aspect of my project involves collecting skin swabs to perform immune gene expression studies using qPCR, which is a similar technology that you have in the COVID tests. This will improve our understanding on the frog's immune response to chytrid infection, and we can also look at the immune response to temperature variation as a measure or as a mimic of climate change. These minimally invasive methods are vital in endangered species where every individual counts and are critical to improving animal welfare in wildlife research. Biomarkers, together with ecological measures, can help us detect stresses in the environment, gauge the success of artificial ponds and optimize conditions in our breeding programs, and can also improve our understanding of impacts of climate change as we monitor biomarker response to temperature variation. To conclude, biomarkers enable us to view populations through the lens of individual animal health and have the potential to be integrated into decision-making for conservation actions for threatened species. Thank you very much for listening today and giving me this opportunity to share my research. And also thank you to the amazing team of people that I work with.
Thanks, Anne, that was fabulous. Again, any questions in the chat? And you've got a lot of claps coming through, so well done. Julie, I might hand over to you.
Yes.
Speaker. Yeah.
Thank you. Sorry, I'm just typing a question in the chat there. Okay, so I would now like to introduce Olivia, and Olivia is going to be talking about safeguarding threatened reef species, and she's from the University of Tasmania. Thanks, Olivia. Oh, is it Olivia there? Yes.
I'm
okay.
sorry, I'm just on different screens.
That's okay, no problem.
There we go. Hopefully, I can share this with you. Can everyone see that?
Yeah.
Yeah. Okay, great. Looks like, does that work on your screen?
So we've got your notes view. You need to pop it on presenter view. That's okay, we've got plenty of time.
Okay, thanks.
Schedule.
All good.
Is that better? No.
Well done. Yep.
Yes. Okay, cool. Fantastic. Sorry. Thank you very much. Good morning. My name is Olivia Johnson, and I'm a PhD candidate at the University of Tasmania's Institute for Marine and Antarctic Studies. Before I start my talk, I would like to acknowledge that I'm extremely fortunate to work on Lutruwita, Tasmania, Aboriginal land, sea and waterways. I'd also like to acknowledge with deep respect the traditional owners of this land, the Muwinina people, which I'm giving this presentation from today. I'd like to give thanks to the Australian Wildlife Society, firstly for providing me with funding towards presenting my research at the upcoming Indo-Pacific Fish Conference in New Zealand, and for inviting me to present and participate in the webinar today. So some brief background on marine biodiversity. Reef ecosystems, both coral reefs and rocky reefs, are some of the most productive and diverse systems on the planet. Australia has extremely rich marine biodiversity, making it completely unique. The endemism is some of the highest globally. For example, 50% of algae species and some of the others listed on the screen are not found anywhere else on Earth. So this results in distinct coastlines around Australia that hold unique biogeographic regions for both rocky and coral reef ecosystems. The oceans are integral to our way of life. They support a range of ecosystem services that have ecological, social and economic implications. But with 87% of our oceans having been modified by humans, they're currently facing a biodiversity crisis. These threats and changes are multifaceted, and they include things such as climate change, ocean acidification, and marine heat waves, just to name a few. When it comes to marine species declines, population trend and distribution data is logistically challenging to collect for marine species. Beyond commercially important or highly charismatic species, few marine species are studied in any great detail. Many species are also rare by nature and are often highly cryptic. So this makes it really difficult to collect data on marine species. Just a couple of facts: incredibly, invertebrates have 1,000 times fewer conservation papers compared to turtles, and compared to birds, amphibians and mammals, as little as 0.1% of marine invertebrates and fish are considered threatened. Rare and endemic species are most susceptible to extinction due to restricted geographic range or small population size, and rapid species declines or rare species can cause overall implications for resource monitoring of those species. Therefore, this makes the extinction risk in marine taxa much higher than terrestrial species. For my project, density data has been obtained from 2 long-term reef monitoring initiatives, which include the Australian Temperate Reef Collaboration, which has been running since 1992, and the Reef Life Survey, which is a citizen science initiative that's been running since 2007. Both initiatives apply comparable underwater visual surveys along 50-metre transect lines, with dive searches applying different methods, typically in 5 to 10 metres, to survey fish, mobile invertebrates, algae and coral species. You can find more about the methods on each of their websites. So for my project, the combined program has over 2 million abundance records, but this was reduced to 100,000 records, or nearly 100,000 records across both the programs. And this comprised of 100 macroalgae species, 21 coral species, 541 fish species, 140 invertebrate species, and this was from up to 465 sites around Australia over the 14-year period. So we conducted a series of linear mixed effects models on the species, just described to understand their population trends. And of the 705 species, we found 130 species with significant population trends. 53% of these species were in decline, and of those in decline, 64% were found to be Australian endemic species, which is shown in this plot on the screen. From further investigation, we also found that 25% of endemics experiencing declining populations would qualify in a threatened category currently listed on the IUCN. Those species are currently listed on the IUCN, but as least concern, and the oldest assessment dates back nearly 2 decades to April 2004. And 73% of the endemics with significantly declining population trends have not had a conservation status evaluated. So this is just a little snippet of what will comprise my first chapter of my PhD, which is hoping to highlight the disproportionate number of Australian endemic marine species in decline that lack any type of conservation assessment and could potentially be heading towards extinction, as well as the need for timely reevaluations of listing assessments from government and IUCN Red List authorities to ensure species are adequately protected. I'd just like to thank my supervisors and everyone else who's assisted me with my project so far, RLS and HRC for the use of their long-term monitoring data, the Ecological Society of Australia for funding me for multiple years of additional field surveys, and most importantly, the Australian Wildlife Society for providing me with funding today to present my results and also at an international conference. Thank you very much.
Thanks so much, Olivia. Julie, would you like to introduce the next speaker?
Yes, thank you. Our next speaker is Hannah Gurke. I hope I've pronounced that correctly. Sorry if I haven't. From ANU, Australian National Uni, and she's going to be talking about movement and behaviour of brown snakes.
That's another pre-recorded talk, so I'll just share my screen now.
Hello everyone, my name is Hannah Girk and I'm a PhD student at the Australian National University. Today I'm going to tell you a little about my research on the movement, behavior and personality of urban adapted eastern brown snakes. I'd like to begin by acknowledging the traditional owners of the land on which we meet. I'd also like to pay my respects to elders past, present and emerging. Now I'm sure you're all familiar with eastern brown snakes as one of Australia's most well-known and perhaps infamous snake species. Brown snakes are both highly venomous and also very capable of exploiting urban areas, which can lead to some tricky situations. Here in the ACT, they're actually the most commonly encountered snake. However, urban areas are a very risky place for snakes to be, and urban snakes are constantly under threat from humans, pets, vehicles, and even our trash, as you can see on the slide. A common response for people when seeing a snake in an urban area is to call a snake catcher, who will then come and move it to a nearby reserve. For a lot of people, that's where the story ends, and they feel pretty good about the outcome. Unfortunately, recent advances in technology have allowed us to actually monitor what happens to wildlife after they've been translocated, and it really doesn't look too good for snakes. In general, snakes have small home ranges, and they're very familiar with where to find resources like food, shelter, and water within that home range. However, when snakes are moved out of the areas that they know, it can be very stressful. Many snakes show excessive or erratic movement behavior after translocation, which makes them more vulnerable to predation and to threats like humans or vehicles. It's also not uncommon for translocated snakes to move back into suburbs, which can lead to further human-snake conflict. In the end, some snakes don't survive being translocated out of their familiar home range. But we're really only beginning to understand the various ways that translocation affects different species and different individuals. So, our main question is, how does translocation impact eastern brown snakes? To address this, we will implant radio transmitters and mini data loggers that measure body temperature into two groups of snakes: resident snakes caught opportunistically at the field site and snakes translocated out of urban areas. We will compare snake survival and growth over a year-long period, track how far and how frequently they move, and measure their ability to maintain a preferred body temperature. This research is part of a broader program called the Canberra Snake Tracking Project, which has already conducted pilot research radio tracking brown snakes throughout the ACT. They've produced some great baseline data for comparison, but we've noticed, even within the pilot research, that there's a great deal of variation among individuals. My particular research will focus on snakes at one field site and attempt to identify some of the causes of differences in individual snake behavior. In recent years, research has shown that a variety of animals exhibit different personalities. Now, animal personality can be defined as consistent differences in behavior among individuals that are repeatable over time. It's often measured along gradients in categories like boldness, exploration, activity, aggressiveness, and sociality. Personality can affect survival, dispersal, habitat selection, disease transfer, reproduction, and many other key processes. Understanding the role that personality plays in individual behavior could help us predict wildlife responses to environmental or human-caused disturbances, and ultimately improve conservation outcomes. For that reason, another goal of this project is to see if we can measure variation in personality among brown snakes, and if we can, test its influence on movement or survival in the field, particularly in regard to translocated snakes. Now you're probably wondering, how do you measure brown snake personality? Well, prior to transmitter implantation, we'll take the snakes back to the lab, put them in a custom-built test arena, and measure variation in behavioral response to a number of tests. We'll measure general activity levels, latency to emerge into a novel environment, the total time spent exploring, and defensive behavior during a simulated attack, among others. We'll conduct 3 to 5 trials per individual and also measure the change in an individual's behavior over the trials, which has implications for an individual's ability to adapt to a new environment or its plasticity. After conducting the behavioral tests, we'll release the snakes, track their movement and survival over the next year, and test the degree to which personality plays a role. Our results will increase our understanding of what causes variation in individual snake movement and provide crucial insight into the ecology of a common but understudied venomous snake. If snakes varying in personality showed differences in post-translocation survival or movement, it could potentially help us improve translocation outcomes and future management recommendations. The support of the Australian Wildlife Society will be critical in allowing us to conduct behavioral tests. Funds will be used to purchase materials for building the customized test arena, as well as cameras for recording snake behavior, so we really couldn't do this without you. I also want to acknowledge the support of our partners below and just say thanks for listening and stay tuned for future updates.
Okay. So Don, if you're there, I might get you to introduce our next speaker. Julie, maybe if you'd like to introduce our next speaker.
Yes, more than happy to. And not that I'm biased at all, but I do love wombats. Everybody loves wombats, don't they? Okay, so our next speaker, is Natasha McPherson, and she's going to be talking about southern hairy-nosed wombats, which I personally just saw recently. I was very excited, my first one in the wild. And she's from the University of Adelaide. Thank you, Natasha.
Thank you. Just let me share my screen. Hopefully everyone can see that there. Okay, so hi, thank you. So I'm going to be basically just discussing a brief overview of my honours project this year that was on the southern hairy-nosed wombat in the Nullarbor. So the southern hairy-nosed wombat is South Australia's faunal emblem, and they are a nocturnal and semi-fossorial marsupial. And much like some of their relatives, they are probably most well known for their construction of extensive underground warren networks that they use for shelter and as a thermoregulatory strategy. Now, the southern hairy-nosed wombat currently resides in 5 main fragmented subpopulations across the state and extends a bit into Western Australia. And although they are relatively stable and abundant at the moment, historical range contractions in response to land use changes, grazing competition, as well as some long-term studies that have noted reduced fitness with prolonged exposure to drought, we want to have a better understanding of species-environment interactions, as well as how populations might be affected in the future and what that might look like. But unfortunately for us as researchers, the largest and most extensive subpopulation of the species resides in the Nullarbor Plain, which is a very remote region with a very distinct, harsh, open semi-arid climate that we have a very strong difficulty in deriving sufficient data for. So my project intended to use a data set that I've been helping to create over the past couple of years, where we've been using warrens as a presence proxy for both the wombat and its largest competitor, the European rabbit. Now, using their distinctive features of these warrens and the open visibility of surface ground in the Nullarbor, we've been able to sample all noticeable wombat warrens within randomized quadrats, where we've derived 33,364 wombat and 7,367 rabbit warren locations, covering approximately 120,000 square kilometers of this region. Now, first, we wanted to be able just to ensure that what we're observing in the satellite imagery is actually the warrens. So we used some field validation, and basically the main takeaway from what we were able to derive, as well as some other data, was that digitizing can account for pretty much all observed wombats, but rabbits were a bit harder to identify, and we had higher emission errors there. But overall, wombats, we were extremely confident in our capacity to digitize. Now, what can we do with this data? Presence data often that we want to use in spatial analysis is small in sample size and often demonstrates a range of spatial biases that violate a lot of basic model assumptions that need to be accounted for during data preparation when we use them for modeling. And so being able to use unbiased data sets that, in our case, describe species homes is really important for us to be able to understand where and why these individuals decide to live, forage, and breed. So just from the extracted data, we were able to note that of the 34 variables that we considered, there we are. Rabbit warrens were distributed across a greater breadth of range for a lot of these, with wombats appearing to be more selective in their warren establishment. But the primary sort of objective was to look at using this data set for species distribution modeling, which allows us to look at sort of a multidimensional measure of relationship between occurrence data and environmental variables, and interpolate sort of a probability of species occurrence or habitat suitability. So we used Maxent presence only algorithm, where we were able to identify that with these interactions, there's a strong association with wombats with pH and mean temperature, while rabbits had a more strong association with rainfall and clay content. And from this, we've been able to derive an idea of how habitat suitability might change in the future according to a range of different climate scenarios. And so this is just sort of a brief visual sort of overview of everything, but at the top we have the current models, and the probability is probably, but the most important thing that probably to take away is that regardless of what scenario that we used or model iteration or settings that we implemented, the general trend that we see is that for the wombats, there's a decrease in suitable habitat as temperatures increase, but comparatively rabbits seem to have an increase in suitable habitat, although this is a little less certain because of their interaction with rainfall, which we find harder to predict for. Now, models aren't sort of definitive, but it is informative for us as a basis of information that we haven't been able to obtain prior, and especially not at such level of detail. And by comparing these models between species and how they interact over time, we can begin to identify potential areas of future concern. And with the consistency of these trends, we want to have a better understanding of how climate change will impact the wombats, regardless of how stable they might appear at the moment. So a lot of the data that we currently use is based on populations that receive a very different climate with much higher rainfall and lower temperatures. So being able to look at this area in detail for the largest population is really critical for identifying that future risk and being able to implement really targeted, effective research in an area that's really hard to access and derive sufficient data for. But yeah, so that was a very brief summary. If you have any questions, feel free to ask. Yeah, it's really exciting. So thank you for the funding. It takes a lot to get out there and it's quite a bit of a drive. So it helps to have a bit of extra funding. So thank you.
Thank you, Natasha. That was great. And yes, it is a long way to go out in the field to see the wombats. It's great. I do have a question, but I'm going to put it in the chat for you. Just a reminder, if you do have questions, put it in the chat. And make sure you send it to everyone, not hosts and panelists, because otherwise not everybody can see it. But we might, at the end, see if we can open up the forum to questions. Okay, our next speaker is Holly, and Holly is from James Cook University, and she's going to be investigating the effects of insecticide exposure and pathogens, poor bees, on bee diversity, abundance and health. I feel so sorry for the bees at the moment. Thank you, Holly.
Thanks, Julie. I'll just share my screen. All right. Hey, everyone. Yes, I'm Holly and I'm from James Cook University on Gimoy country, Cairns, and I'd like to start by acknowledging the traditional owners of the land on which I stand. So I'll be talking about my PhD research, investigating the effects of insecticide exposure, pathogens and parasites on bee diversity, abundance and health. So when you think about bees, probably the honeybee pops to your mind, but in Australia, we actually have over 1,800 species of bees. And in the world, we have over 20,000 species. And most of these are actually solitary, which means they don't live in a colony. Usually it's a female bee and she'll provide nectar and pollen for her babies and she'll build a little nest for them. And so these bees are very diverse in size, form, colour and behaviour. And this is just a few of the beautiful bees that we have in Cairns. But unfortunately, bees are under threat, and this is due to multiple stresses, including insecticide exposure, pathogens and parasites, poor nutrition, climate change, introduced species, and loss of habitat. And so my research will focus on insecticide exposure and pathogens and parasites. So both of these stresses have sublethal and lethal effects on bees, but limited studies have looked at the combined effects on non-apis bees. And a non-apis bee is any bee that's not a honeybee. So bees are exposed to multiple stresses throughout their life, and the combined effects of these stresses could be synergistic, which means they could add on top of each other and make the effects worse. So the first aim of my research is investigating whether insecticide exposure is associated with decreased bee diversity, abundance, and functional diversity. And to do this, I'll be conducting a field study in 2 contexts. The first will be rainforest treated with insecticide for control of invasive species. And the second will be rainforest adjacent to sugarcane treated with insecticide for control of pests. So I'll use complementary sampling methods, including malaise traps, vane traps, and trap nests, and my field study will run for a year. So my second aim is investigating whether insecticide exposure is associated with decreased bee predator abundance and pathogen and parasite prevalence. And so the insecticides used in these contexts are non-discriminatory. So they could affect any organism in the ecosystem. And this could include bee predators like wasps or kleptoparasitic bees, also possibly skinks or ants or mites as well. And then my third aim is investigating the effect of nesting density on bee predator abundance and pathogen and parasite prevalence. And so you might have heard of bee hotels or insect hotels or pollinator B&Bs. They come in lots of different shapes and sizes, commonly sold at Bunnings or online. So these hotels utilize some bees' nesting preferences, and they're often claimed to be good for bee conservation. But they artificially aggregate nesting sites above densities naturally available for cavity nesting bees. And this could possibly increase pathogens, parasitism, and predation. So I'll be conducting an experiment in the field to test this, and I'll be using 2 designs of bee hotel. I'll have an aggregated design, which will be 12 blocks clumped together. I know that's only 9 in the picture, but I will use 12. And then a dispersed design, which will be the same type of blocks, but they'll be dispersed across a grid, each 5 meters apart and attached to individual trees. And these are some of the design features, and they'll be checked every 3 months. So once a bee nest is capped off in the field with resin or leaves, I'll be able to tell that it's a bee. I'll remove the straw that will be inserted inside the hotel and take it back to the lab, and I'll rear it until an adult emerges, and I'll also dissect the nest contents to extract DNA and RNA. And then I'll test for fungal parasites, bacterial pathogens, and common Apis mellifera viruses. And this part of my project was very generously funded by the Australian Wildlife Society, so thank you so much for that. And on the right, we have some of the pathogens that I'll be looking for. So far, I've been very busy building hundreds of bee hotel blocks, and I've also piloted my methods for checking, and I've had a few bees visit. This is a fire-tailed resin bee in this picture here. And then I've also sampled some bees in my vane traps, and I'll be starting my field study in the next few months. So stay tuned to see what I find. Thank you.
Thank you, Holly. That was great. Poor bees are really copping it at the moment.
I know.
So I will introduce our next speaker. That is Tina Now from the Invasive Species Council, and she's going to talk to us about cat management in Australia.
Hi everyone, I'll just share my screen. Is that working?
Yes.
Great. It's not showing up for me for some reason, but I'll roll with it.
We can see your wonderful notes.
Oh, okay. How's that?
I can't see anything.
Oh, gosh. I'm failing today. It's showing up for me. That is so weird. Okay.
That's okay.
Oh, hang on.
That's all right. While we're waiting for that, I was going to ask a question. What else can we help? What else can we do to help the bees, Holly?
I just started responding in the chat, but I may as well say here. So the best thing that we can do to help our bees is to plant native plants, preferably native to your area. You can always ask at your local nursery. And also maintaining natural habitat. If you have like a tree fall down, it can be really good to leave it as a nesting space. And bees also require a safe environment, so try not to use pesticides in your garden, and also just spread awareness and look out for like events like this and have a conversation with people, because lots of people don't realize that we actually have a lot of bees other than honeybees.
I've got to say, I have got the wonderful Bunnings bee hotel. And I think maybe I've had one. Is it something about you've got to put in a special position or something?
Yeah, so there's a lot of different factors. Some of the Bunnings hotels, the reeds or the bamboo actually aren't deep enough for bees. So native bees require cavities around 10 to 15 centimeters long. So actually quite deep, and they will go all the way in and then build lots of different nest cells. And they also like to have a nice shady spot, so somewhere that doesn't get blasted with morning sun, a bit sheltered. But yeah, I've also got a Bunnings hotel in my backyard, and I've only found wasps in it so far. Yeah, hopefully my research will be able to come up with some better alternatives that are more suited to native bees.
Yeah, that's great. That's great. Oh.
And
here we go.
Yeah.
Is that working now?
Go ahead, I'll keep answering questions in the chat.
Thanks, Holly. Yeah, we can see it, but we've just got your notes. So yeah, maybe just click from the beginning, see how we go.
How's that?
Yes, excellent.
Okay, cool. Excellent. Sorry about that, everyone. Hi, thanks so much for joining us today to celebrate Australian Wildlife Week. My name's Peter. I'm joining in from the lands of the Yagara and Turrbal people. I'd like to acknowledge the traditional owners of the land, waters and skies, which are home to our amazing array of native wildlife. I pay my respects to elders past and present, and to the rangers who care for our country. Today, I'll give a quick overview of where we're at with cat management in Australia, and I'll briefly describe how cats fit in as a threat to Australian biodiversity. I'll do a quick snapshot of how cats fit in, how cats are managed in each jurisdiction, and highlight the key strengths and weaknesses, and finish off with an opportunity to improve cat management in Australia. So on a global scale, the biggest threats to biodiversity are overexploitation, agriculture, and urban development. But here in Australia, the story is a little bit different. Invasive species followed by ecosystem modification, particularly changed fire regimes, and agriculture are the most prevalent threats to Australia's threatened species. This is because of Australia's geographical isolation. That's thought to be one of the main reasons that our wildlife is particularly susceptible to invasive species, in particular foxes and cats. But in this talk, I'll focus on cat impacts. Cats in Australia have a much more severe impact than in other continents, and this is thought to be for three key reasons. Firstly, Australian wildlife has evolved without any cat-like predators. Secondly, because our native species have low rates of reproduction, so their viability is readily compromised by predation. And thirdly, because cats are pervasive in the landscape and cats are such efficient predators. It's well known that Australia is a global leader for all the wrong reasons. We have the highest rate of extinctions in the world, with at least 34 mammals becoming extinct since 1788. This includes species like the pig-footed bandicoot, white-footed rabbit rats, the desert bilby, and 4 species of hopping mice. Cats have played a primary role in 28 of these extinctions, and cats are a primary threat to our native wildlife. This includes our beetles and butterflies, arid zone reptiles, ground nesting birds, frogs as well, mammals weighing between 35 grams and 5.5 kilos, so including our mulgaras, our greater stick-nest rats, and barred bandicoots. I'm not going to go into detail about the varying concerns of impacts of cats. I'll let you read this slide for yourself, other than to say that there have been concerns expressed by early naturalists, Indigenous rangers and conservation scientists, and increasingly amongst community members across Australia. So there's a lot of information on this slide, sorry for that, but the key points from this slide are that there are an average of 2.1 million feral cats across Australia. It varies according to climatic conditions, and they occur over 99% of the land mass. They inflict a substantial toll on Australian wildlife, and they're difficult to control because they're wary and they prefer to eat live prey. Oh, sorry. There's also an estimated 700,000 stray cats in Australia, and management of these cats presents a significant challenge. It typically falls to local governments, which might not have the resourcing to manage them well. Thirdly, with the increase in pet ownership associated with COVID-19, there's now an estimated 5.3 million pet cats in Australia, 71% or 3.7 million of which are allowed to freely roam. I'll now do a quick run through of cat management practices in Australia and highlight any key strengths and weaknesses in each jurisdiction. I'll use a quokka to denote a strong action, so it may not be perfect, but it represents a step in the right direction. And for weaknesses, I'll have a little cat icon. This denotes that there's weak legislation which hampers effective management of cats. So the ACT has 24/7 cat containment in place for all pet cats born or after the 1st of July 2022, with grandfathering arrangements in place for cats born before then. They also have 17 cat containment suburbs in place, as well as early age desexing, which is a really important measure for preventing unwanted litters before they're born. Also safe and recommended. New South Wales. So New South Wales has really weak companion animal legislation in place. This basically allows pet cats to freely roam, except in wildlife protection areas and food preparation areas. The legislation doesn't allow local governments to set local bylaws, so essentially the state level legislation hampers effective management by local governments. Queensland is home to a high proportion of our native wildlife, but there is no strategy for the management of feral cats. There are some dedicated efforts by the Queensland Government Department of Environment and Science to reduce the risk of predation to greater bilbies in Astrabli National Park and to bridled nail-tail wallabies in Taunton National Park. These efforts are commendable, but it's also really crucial that they get local governments on board to make sure that there's integrated management of both pet cats and feral cats. I'll just quickly note that there is a bounty bonus payment in place in one local government area, but these are typically ineffective. Moving on to Victoria. Victoria's management of pet cats spans the spectrum of no restrictions, dawn to dusk curfews, and 24/7 containment. In terms of feral cats, they've been declared as a priority pest animal, but this only applies on Crown land. Victoria has a pretty poor record in biodiversity conservation and is currently developing a cat management strategy which will support education programs, grants to support cat containment and to promote responsible cat ownership, but we really don't know what the implications for feral cat management will be. Similar to the rest of Australia, the management of cats in Tasmania is highly variable, with the most concerted efforts in place being in Kingborough Council and Bruny Island, where there's dedicated funding in place. Of note, there is a Tassie Cat program in place, which also provides a centralised source of information that encourages responsible cat ownership and a level of regional coordination for cat management. Cat management in South Australia is similarly patchy. There are common reports of cat overpopulation across South Australia, and there's been some efforts to address this issue by animal welfare organisations. Local governments can set local bylaws, so that's an important thing, but local councils don't always have the compliance and enforcement powers they need. Local governments have also attempted to put some cat containment measures in place, but at times they may face criticism and backlash. There are plans to develop a statewide approach to cat management and also some dedicated biodiversity legislation, so that'll be good to see, but will take time. The Northern Territory is the only jurisdiction in Australia which does not have companion animal legislation in place. So this is an important mechanism which can foster responsible pet ownership and provide an overarching framework for local governments to work with. The absence of this in the Northern Territory means that local governments are on their own, and so there is a risk for any local government wishing to be proactive on cat management that the problems can be shifted from one LGA or local government area to another. In terms of feral cats, there are some cat control programs in place, particularly to protect the critically endangered central rock rat in Central Australia and for a range of species on the Tiwi Islands. Lucky last is Western Australia, which has been the most proactive jurisdiction on managing feral cats. This includes the successful eradication of cats from 9 islands and managing cats to protect numbats, banded hair wallabies, golden-backed tree rats and the rufous hair wallaby. Unfortunately, WA is let down by its legislation for pet cats, which does not allow local governments to set local bylaws for cat containment. I just briefly wanted to touch on remote Indigenous communities where in the last 10 to 15 years, pet cats have become increasingly common. But being remote, there's really limited access to veterinary services and rapid growth of cat populations has been documented in multiple remote communities. There are some organisations and veterinary service providers which provide services to these areas, and continued support of these services is vital as in terms of stabilising cat populations and dog populations and reducing the level of transfer of cats in particular to the feral population. Now, to wrap up with an opportunity, there is one right now. Anyone with a love of native wildlife can get on board. The 4th iteration of a threat abatement plan for feral cats is currently open until the 11th of December. We encourage you to review the plan, which is very comprehensive, and if you wish, have a say. We've also listed this as a threat, so unfortunately there is no obligation under Australian environmental legislation to allocate funding to threat abatement plans. If this is the case, then it'll be business as usual and cats will continue to have a significant toll on our native wildlife. But if it's fully funded, then we can start to turn things around. To wrap up, I'd just like to encourage you, if you haven't already, to visit a predator-free sanctuary. These places are small microcosms of Australia as it used to be, with a diversity of tracks on the ground and the little sounds of ruffles and scruffles and hopping that you don't normally hear when you're out in the bush. And you can also have an opportunity to see some of the species that are most vulnerable to predation by cats and foxes. So as you can see from the quick snapshot of cat management across Australia, it's quite disjointed, it's not well coordinated, sometimes non-existent and sometimes ineffective. the feral cat threat abatement plan is an opportunity to turn this around. So if you want to get on board and have a look. Thank you, and thanks to the Australian Wildlife Society for their support and for inviting me to be a speaker today. Thanks.
Thanks so much, Tita, and we appreciate you having you on board, chatting about cats. And I just wanted to point out that the Society will be making or commenting on the threat abatement plan. And there's a few directors who have pet cats as well. So we do love our cats, but we also adore our native species and have a priority to ensure that they are preserved and protected for future generations. So we'll be putting all of those in the comments and hopefully getting a successful outcome for the threat abatement plan. So thanks again for all the great work that the Invasive Species Council is doing.
Fantastic work, fantastic work.
Over to you, Patrick. Thank you.
Right, thank you again. What an amazing group of speakers, all focused on wildlife conservation right across Australia. It's my pleasure now to introduce the next speaker. Megan, you'll have to remind me.
Yes, so we've got Ocean Atlin from the University of Western Australia.
Wonderful. Right, thank you. Well, over to you. Over to you. Thank you.
Hi everyone. Has been a great morning. A lot of different talk, and I've learned a lot. That was great. So I'm very happy to be to share. So I'm going to share my screen and to share with you what I've been working on. Can you see it? Yep.
There we are. Wonderful.
Oh, good. Is it better like that? All right, great. So I'm going to bring you on a journey, an underwater journey on the Western Australian coast. So I am studying Sunbird Reef on the west coast. So I am part of the university. So I'm doing a PhD at the UW University of Western Australia. And so I would like to start my story by describing what is on this picture there. So I've been taking this picture in Perth last summer, and you can see the two main organisms that I'm going to talk about during my talk. So you can see at the background those brown kelps, so Eclonia radiata, which are seaweed that are dominating Western Australian Sunbird Reef. So they create a very complex habitat where fish and other type of organisms, just like crustacean and other invertebrates, rely on as an habitat or resources, where they can find food or shelters. And then you can see at the front those huge coral colonies. So that's my two main actors actually of my story there. So to begin with, we have seen huge changes in our ecosystem here in Western Australia. So the temperate reef goes as far north as Kalbarri, which is 7 hours north of Perth, and goes all the way down to New South Wales, actually. So that's a huge ecosystem, continuous ecosystem. But lately, we have seen dramatic changes, especially at the warmer edge. So for example, in Kalbarri, after the marine heat wave in 2011, most of our kelp have completely gone now. And so they are replaced by other type of ecosystems, such as tropical seaweed that have a very different function than the kelp forest. And where I'm going to spend a bit more time is like some of our dive sites in Port Gregory, which is like only 40 minutes south of Kalbarri. We have seen huge changes in the ecosystem where we have turf states. So another panelist was talking about turf states in the East Coast. So we have the kind of same thing here in Western Australia, but also some dive sites are actually dominated by those huge corals now. And we don't know much about those corals. We don't know how much do they grow, if they grow as fast as tropical coral, for example, if they have a different growth rate according to seasons. So that's something I'm trying to study for my PhD. And so this is very important because the coral will completely shape the ecosystem. So they will shape the complexity of the rocky reef. Is going to create more sediment and another type of habitat for associated species. So for the last year, I've been going in the field. So I've been collecting some corals. So I have different species that I've glued on ties, as you can see. And then I have this frame underwater where I just leave the corals. They settle there and they just slowly calcify, slowly accumulating calcium carbonate. And then after each season, I am able to weigh them, assess the growth and how much they accumulated this calcium carbonate. So I've been doing this in Port Gregory, but I've been also having this project in Perth because we still have a coral community, but it's very different than the one that we have in Port Gregory, but it's still to have an idea of how much those coral are going to calcify over the seasons. And so thanks to the Australian Wildlife Society, I've been able to go a bit more in the field and I will be able to keep going on this project. So now I want to go at different locations. As you can see on the right on my screen, I have like different locations where I want to assess the coral community and how much they shape actually the rocky reef. So it's a method that they use in tropical reef where they can assess the rugosity of the reef and measure how much the complexity of the reef is. So that's what I'm going to do for the next season. Summer season is coming here, so that's going to be a lot of field work for me, and so I'm very excited about it. And yeah, I hope I will have good results that I can talk through later on. So I think that's pretty much it for me. So I'm just going to say thank you for all the funding and for to the Australian Wildlife Society as well, and a huge thanks for the Wember Lab where I'm part of. So big thanks to like all the people that can help me on the field, because I'm not the only one. I cannot dive by myself, so I need a lot of help, a lot of divers as well with me. So that has been very, very nice the last season. So hopefully the next season will be as good. Thank you very much for listening.
Fabulous. Thank you so much, Ocean. And keep up the great work. We have a lot of claps coming in for you. So well done. So I'll get Brian to introduce our next speaker, if you please.
Hello, I'm delighted to introduce our last speaker for today, Dr. Kit Pentecast, one of the Society's 2022 Conservation Grant recipients, who will speak about something that's very close to my heart, native bee conservation. Thank you, Dr. Kit.
Thank you so much for inviting me to speak at your wildlife seminar. And firstly, a big thank you to the Australian Wildlife Society, who have helped fund quite a bit of my research, and I'm going to be talking to you all about it. So firstly, I'm just going to try and share my screen. Here we go. So I'm going to be talking to you about native bee conservation as a whole in a very short time. So let's get started. Firstly, native bees. Now, most people learn about bees in terms of the European honeybee, which is an introduced species in Australia. But we have an incredible diversity of native bees. We have over 2,000 species, and where I conducted my research was in Western Australia, where there's about 800. And most species in Australia are endemic, which means they're found nowhere else in the world. And these are estimates of the species diversity. So there's actually 1,661 described species, but we have hundreds more that are undescribed, which is really exciting, but also a bit scary, because there are so many species that have not been given a scientific name. And if we don't know something, we can't study it, we can't protect it. And that's the next issue. Only 4 of these species are given legislative protection, which is a very small fraction. And this is, I guess, goes for invertebrates as a whole. But there is a lot of work to do in native bee conservation. Now, native bees as well are so important for conservation as a whole because of their roles in ecosystems. Now we have 5 out of the 7 global bee families. We've got Apidae, which includes the same family as the European honeybee, but we've got the Amygilla, or sometimes called blue-banded bees. There's also Amygilla dorsoni and the teddy bear bees. We've got the Megachilidae, which are some of my favourites, and the Australian Wildlife Society helped me write up a publication all about the Megachili and the impact of honeybees on them and their use of bee hotels. We have the Halictidae, which are ground nesting bees, the Coletidae, which is our most diverse family, and the Stenotratidae, which are an entire family that is only found in Australia. And they include that beautiful metallic green bee down the bottom there, Stenocoletes smaragdinus. So incredible diversity. As you can see, they all look very different from honeybees, and their ecology and lifestyles are also very different from honeybees. So protecting honeybees doesn't protect our native bees. Most of our bees are solitary. They don't live in colonies. They don't care for their offspring. We've got some semi-social ones. We've got eusocial ones, like the Tetragonula and Ostreoplevia, which are the sugar bag bees that are important part of Indigenous people's culture. Our bees range in size from some of the smallest in the world, 1.9 millimeters long, to some really big ones like the Xylocopa coptortozoma and Amygilla dorsoni. So huge size range. Some of them are very furry, other ones lack hairs completely. So very different from honeybees. Now, a key thing to conserving our native bees is, of course, planting flowers. But what my research has found, that planting any flowers actually won't help our native bees. Our native bees are very specialized. So I've got a publication there. Native flora receive more visits than exotics from bees, especially native bees, in an urbanized biodiversity hotspot. So I conducted my PhD research in the southwest of Western Australia, and I was researching people's gardens as well as bushland remnants and looking at what flowers the native bees were collected from. And so many native bees exclusively foraged on native plants. Many of our native bees are very specialized. They will only forage on Myrtaceae or only forage on Fabaceae. So even if you have a garden that is full of flowers, it can essentially be a desert for our native bees. And I found actually that the more flower species there were in the garden, the fewer native bees there were, which goes against, I guess, conventional common knowledge, but that's because our native bees are specialized. And we know Australia's a continent that's been isolated from other continents for hundreds of thousands of years. And you know, if we just look at our marsupials, weird wildlife that have their own ecological niche that do things a bit differently to animals in the Northern Hemisphere. Same with our native bees. So high proportions of native flora, in particular the Myrtaceae and the Fabaceae, can't go wrong with those. And as you'll also notice, these plants in these families are trees and shrubs. So I guess against what we learn in, I guess, for honeybees plants lots of like sort of ground dwelling flowers, lots of our native bees need those big flowering trees. So we need to keep trees, we need to keep those street bird trees, and yeah, plant lots of natives. But our native bees don't just need food, they also need somewhere to live. Unlike honeybees, they can't be managed in colonies except for the sugar bag bees. The majority of our native bees, they are wild, and we can't just put them in a hive. So a large proportion of them nest in pre-made cavities in wood created by wood-boring beetles. So we need to have intact ecosystems. We need to keep our trees, but we can also help supplement these resources with bee hotels, but not just any bee hotels. We need to make sure these are designed correctly. The hole diameters need between 3 millimeters and 10 millimeters. They need to be at least 100 millimeters deep. We should use wood that hasn't been treated with pesticides or fungicides. And we can create bee hotels by drilling holes in wood or getting bamboo and stacking them together and installing them in sunny locations, but not direct sun, at about head height. And I have published a paper all about the use of bee hotels by native bees. Some native bees love them, other ones won't use them. So you actually have a different proportion of bees that you see that can use them in the field versus the cavity nesting bees that are flying around. But they are a very useful tool. And I have a paper that is under review where I was able to look at the pollen contents inside the bee hotels, rear hundreds of thousands of nests, and look at that in relation to the honeybee densities in the area. So they're actually really good at not looking at just how honeybees affect how many native bees are foraging in an area, but the honeybee impacts on fitness, that is the reproductive output of the native bees, which is, of course, the key thing. They need to be able to reproduce. So yeah, stay tuned, because that's the paper that the Australian Wildlife Society helped me write, and it's under review, so hopefully it'll be out soon. We've also got ground nesting bees, so we need to leave patches of bare soil. Of course, not put fake grass down, minimize tilling our garden beds. And of course, in urban areas, limit putting down lots of pavement. Pesticides, we don't need to use them in gardens. Targeted next generation pesticides can be good if it means that we have to clear less native vegetation. Herbicides are, again, mixed bag. If we're using them to clear invasive grasses or invasive weeds, they can be good, provided that we plant native plants so that it's not just nothing left for the bees. So yeah, limit pesticides is, I guess, the take-home message, but if we are using them, use them in a conscious way so that we can actually help boost native flora for our native bees. Animal agriculture is a major issue for biodiversity as a whole, as I'm sure we all know. And in addition to massive land clearing of rainforest, animal agriculture is the main cause of land clearing in Queensland, which is where I am now. But it is also a major contributor to climate change. And this is something that I have published a chapter on. And it disrupts the synchronicity between the activity periods between bees and flowers. Places are now getting too warm for the bees and the plants. We see animals and plants migrating, but because bees are so closely tied to their floral resources, they need to migrate at the same rate. In Australia, I guess the main effect we're seeing of climate change is droughts and bushfires, which of course have major impacts on our native bees. So, disrupting the nectar flow, destroying habitats, and also the increased carbon dioxide in the atmosphere is altering the nectar and pollen chemistry. So many, many impacts here of climate change and land clearing on the native bees. But the good thing is that everyone can contribute to reducing this threat by reducing your meat intake. So going vegetarian, you cut your carbon footprint by half, and many other benefits to biodiversity as a whole, especially here in Australia, where our landscapes have not evolved with hoofed animals. Now, to understand how our native bees are doing, we need to be monitoring their populations, and this is not happening. Most native bees, we don't know, we don't have long-term records, that we can sort of do space for time comparisons instead. But you need to use the right monitoring methods. And so this was a core part of my research as well. And I was comparing passive methods, putting out bee bowls, vane traps. These are coloured, essentially, traps that attract the bees based on colour. And then I was also going out and actually collecting the bees. A big finding was that you actually need to go out in the field and collect bees, which for someone like me, I'm very happy to do, but it does mean a lot more person power and a lot more expertise than putting out coloured pan traps. But the issue with these is that the coloured pan traps, they collect more bees in bad habitats. And so there's actually been studies that have been published where they've just used these pan traps and have said bees are doing better in, managed agricultural landscapes and in native bushland. But if you go to these habitats, that's not what you physically see. It's because in bad habitats, when there's not many floral resources, the bees are going to go to these pan traps rather than go to the flowers. So we definitely need to make sure that when we're doing any bee monitoring, we need to use the right methods. And another big issue is that we are severely lacking in taxonomic investment and expertise in Australia. As I said, we've got hundreds of species that are undescribed, and that's because there is actually no taxonomy courses or degrees in bee taxonomy or in invertebrate taxonomy in Australia, and there's no investment in taxonomy. So being able to identify species is, of course, at the core of any biodiversity assessment. We need to know how many species there are in an area, but identifying bees is very, very difficult because their diagnostic features are microscopic. Many genera don't have keys. The descriptions were hundreds of years old. And so we've got a lot of work to do there in that regard. But taxonomy is, I guess, at the very base of any biodiversity assessment. And so we need to really get that right to properly understand and conserve our native bees. So yeah, we need to invest into taxonomy and also make sure that when people are reviewing any papers on pollinators, they're aware of the limitations of the methods that are being used. So another issue is honeybees. Introduced species. Honeybees are getting a lot of press at the moment because varroa has recently been found in Australia for about a year. It is a threat to honeybees. It's not a threat to native bees, fortunately, but some of the eradication measures, such as putting out toxic bakes, could be. But honeybees themselves are a threat to native bees. And so varroa might help our native bees actually get a bit of the leg up. And I have been researching specifically how honeybees impact native bees, and they do cause competition for floral resources for the native bees that are specialised and have a high resource overlap. And they also impact the pollination networks. So this is the plant-pollinator interactions, and they change how robust these networks are and how connected they are, making them more vulnerable to disruptions. So if anyone wants to read more about that, those are my papers. So sustainability of beekeeping is something that we need to be really careful about. Getting a honeybee colony in your backyard is not a solution to helping the bee problem. It will actually exacerbate the problem of bee conservation. Even with the sugar bag bees, these are the native ones that you can keep across the north of Australia down to Sydney. We do need to make sure that these are managed sustainably as well. And I think we just had to talk about, cats and dogs. People have to register their cats and dogs. I think honeybees as a domesticated species, we should be registering them because they are also like, cats, a species that when they go feral, they can cause a lot of problems, not only for our native bees, but for our native possums and our native birds. So as I talked about, taxonomic impediment is something that is facing invertebrate conservation as a whole. There are hundreds of species that are undescribed. Many genera don't have keys. And with the advent of DNA barcoding, we're also finding that we have to do a lot of revisions because species have been described a number of times and it's actually the same species. I described the most recent bee, 1661st species, Leoproptis zephyr. I named it after my dog. It's a very cool bee. No conservation legislation for it, no recovery plans. It's only found in 5 places, forages on 2 species of plants. If this was a mammal, it would be critically endangered. Everyone would be looking out for it because it's a native bee, unfortunately, not so protected. We need to invest more in niche topics like the natural history of species. Our native bees, so many, we don't even know what they forage on, where they nest, what their nesting substrate is, what their limiting factors are. And yeah, we need greater incentives for people to get into native bees, to publish research, and they need to be part of any sort of impact assessment, because if you want to clear habitat, you do need to look out for koalas. You don't need to do any assessments of how many native bees there are in the area. And of course, we need to actually get our native bees some legislative protection. Devil in the details, I guess, is the main overall summary of my research. Plant flowers, but we need to make sure we know what flowers we're planting and what sort of bees need those particular flowers. Increasing populations of native bees, if they're sugar bag bees, they're also managed, so we need to make sure we do that. Sustainability, outright ban on herbicides, probably not the best idea in some contexts. Installing bee hotels is a good idea, provided that they are designed correctly. The great thing about native bees is that people can contribute to their conservation, because they can be found everywhere in your backyard. Almost certainly you have native bees in your backyard, especially if you live in Perth. But they are around everywhere, our suburbs, and once you know what to look for, you'll start seeing them. And you can contribute to the monitoring of native bees. So I've got a citizen science project on my Facebook group, The Buzz on Wild Bees. And so you can go there and learn more about native bees and how to identify them. I've got a book as well on how to create bee hotels and a big list of some of the best plants to plant for your native bees. So you can check that out as well. That's my email there if you want to get in touch. And also I've created a little guide on how to identify bees, because as I said, there's no taxonomy courses. So I'd love everyone to start learning how to identify bees. And it's really fun. It's like being a real life, Pokemon catcher, I guess. And I've got some little bingo cards as well to help people learn what bees there are in your area.
Thank you so much. Sorry,
yeah,
we
sorry,
might have to
I'm
leave
going
it there.
over time.
Too much, sorry.
Okay.
But yes,
so thank you, everyone.
Definitely contact Kit if you want some more info on bee. I know there are some questions in the chat there that you might want to look at. Kit, thank you very much. And we're nearly out of time. So if anybody does have any questions, they might be able to very quickly put it in the chat and see if everybody can answer. Otherwise, I just want to thank everybody for speaking today and participating as part of Australian Wildlife Week. It is fantastic that there are so many people watching us on YouTube and Facebook from all around the world. It's absolutely fantastic, and we hope that that helps get information out about Australia's flora and fauna and increases that understanding about natural ecosystems and conservation. So if you do want to find out any more about today's project, you can go to our website, www.aws for Australian Wildlife Society.org.au. And you can also drop an email, and the wonderful Megan will be monitoring that. And that is info@aws.org.au. And finally, if you're not already a member, please join. All contributions to the society go towards supporting both students to do research and organisations dedicated to in the field conservation of plants and animals. So you can check out more information if you would like to join as a member or to donate to the society at aws.org.au. Thank you very, very much.